Today I will talk about large trials, so late phase, when we may have some subgroups of patients that may respond better to treatment. It is based on a real clinical trial as a motivating example. So it was in non-small cell lung cancer, which is the most common type of lung cancer. It's approximately 85% of cases. And the major risk factors are smoking, age, genetic predisposition, and environmental exposures. And the standard treatments, it really depends on the stage of the disease, of the molecular profile, and the overall health of the patient. Despite current therapy, many patients relapse, and treatment response is in fact inconsistent. And in this real clinical trial, we had a new perioperative treatment that I will call drug D plus B, combined with chemotherapy that we want to test in a clinical trial. Just some information about this new treatment. So we thought that we had opportunity to have a big impact on the disease because we had the opportunity to increase cure rates with this perioperative approach. We still have high unmet needs because only approximately 20% of PCR pathological complete response rates are observed. And we know that we have high rates of recurrence for patients who do not achieve PCR. Biological heterogeneity in response to experimental treatment suggests that level of biomarker expression may predict better treatment outcome. So in this context, we discussed a lot, and we were very uncertain about should the treatment response on the overall population or only on the biomarker positive patient. It's a drug for which we had large commercial opportunity, but a competitive landscape. So why should we focus on subgroups? We know that not all patients respond equally to the same treatment. So in non-small cell lung cancer, we know that there are some patients with specific biomarkers that may have significantly better outcomes. And identifying the subgroups allow for targeted therapy to improve efficacy and to reduce unnecessary toxicity. So we can have a clinical impact. We want to reduce the adverse effect in non-responders, and we want to optimize the therapeutic outcomes for responders. So just to explain you what will come after, because after we will compare different statistical strategy, and just for you to understand why we are doing all that, I will just introduce quickly some main statistical aspects of late phase trial. So we have a different type of error. So let's imagine that our drug is ineffective. If our drug is ineffective, we want to control the risk of putting an ineffective drug on the market. So this will be what we called in statistics or type 1 error control, which is something really important because, of course, we don't want to release on the market an ineffective drug. So we want to control this error. And usually for late phase trial, it's at 2.5%. And in the same times, there is a type of correct decision, which is we want to maximize our chance of releasing on the market a drug that is effective. So our big constraint is this type 1 error. We must not go over 2.5%. We really do not want to put on the market an ineffective drug. And under this constraint, we want to maximize our chance of releasing an effective drug. But here we are dealing with the whole population, but also with subgroups. And if we are testing both, in fact, we will increase our risk of putting on the market an ineffective drug. And for you to understand why, I will take a comparison. Let's imagine that you have a bag here filled with 40 sweets, and 39 of them are delicious pink sweets, but you have in the bag one disgusting, awful tasting sweet. And you will do the experiment, you will reach into the bag, you will mix it well, and you will pick one at random. If you do that, you will have 2.5% chance of getting the disgusting sweets. But then if you play several times this game with a complete bag, the more times you play, the greater your cumulative chance of eventually tasting that one nasty sweet becomes. So in clinical trials, it's the same. So playing several times, meaning testing here several population. So if you play several times, if you are doing more tests, it's similar. You will increase the risk of releasing on the market an ineffective drug. And so what I will discuss just after is different strategies to control this risk and maximize our chance of putting on the market an effective drug. So we will be in the context of a seamless phase 2-3 clinical trial. So I'm lucky all these concepts were already introduced by other speakers. Thank you. So we will combine a phase 2, which evaluates efficacy and safety, with a phase 3, a confirmatory trial of efficacy, into a single trial, which enables to reduce trial duration and also patient recruitment. And in this context, at the end of phase 2, we can make some adjustments. So it could be sample size re-estimation. It could be dropping some arms if we have several doses, for instance. But here, we will be in the context of enrichment design, where we may want to select some subpopulation. And of course, we are doing all that considering regulatory requirements to ensure that to maintain our trial integrity. So now I will develop what is our motivating example. So it's a phase 2-3 perioperative trial in non-small cell lung cancer. So our patient population were stage 2A, 3B resectable non-small cell lung cancer. We had two treatment arms. So one arm here, where in the neoadjuvant part, we have our new treatment plus chemo. And in the second arm, we have a control plus chemo. And in the adjuvant part, we just remove chemo. We had a different endpoint for primary endpoint for phase 2 and phase 3. So for phase 2, it was pathological complete response, which is defined as the absence of viable tumor cells in resected tissue. And for phase 3, it was EFS, so event-free survival, which is time to recurrence, progression, or death. And in this, so as I told you earlier, in this real clinical trial, we discussed a lot with the clinical team, and they thought that there may be some subgroups that may respond better to treatment. So we had one biomarker. So thank you. It may be one predictive biomarker. The prevalence of this predictive biomarker was 60%. And from the discussion, they really didn't know if the whole population is responding or only the biomarker positive subgroups will respond to treatment. But what we assume is that biomarker negative should not perform better than biomarker positive. So it's higher, either full population or biomarker positive. So as I said, in phase 2, we will assess a short-term response, PCR, after 6 months. At the end of phase 2, we consider a futility analysis to stop the trial earlier if we think it's futile. And we may consider also enrichment strategy at the end of phase 2, so I will develop that a bit later. And our phase 3, in our phase 3, we consider 2 interim looks for efficacy, so at 50% and at 75%, to stop the trial earlier if we have a overwhelming benefit. From the discussion with the sponsor, they say that claim on both population will be appreciated. And then our challenge was which design, which strategy should we prefer in this context. So what we did is that we compared different strategies and see if we can make some general recommendation on what we should do, knowing that we were uncertain about the response on the overall population of the subgroups. So the first approach we compare, it's the traditional approach. We just consider the entire population. So we don't consider subgroups at all. But of course, if there is only a subgroups effect, we will miss it and we will have a diluted effect on the overall population. Then another strategy will be to consider subgroups analysis. So there are different strategies for subgroups analysis. We consider hierarchical testing. So it's just we test one group and then another. So for instance, we test first the patients in green that are biomarker positive. And if we have an effect on this group, then we can test the full population. And on the contrary, we can also test first the full population and then test the biomarker positive if it's significant. And we also consider other strategy where we test simultaneously the two populations, so full population and biomarker positive. And of course, as I said earlier, we make some correction to control the risk of putting on the market an ineffective drug. And finally, we also consider some enrichment strategy. So enrichment strategy meaning that we may only continue the trial with the subpopulation. So at the end of phase 2, I told you that we may want to stop for futility. If the biomarker negative patient responds, we assume that then a biomarker positive patient should also respond. So we will continue with the full population. But if the biomarker negative patient do not respond, but the biomarker positive patient responds, then we'll continue the trial only with those patients that benefit the most here to the treatment, and we will stop recruiting in the whole population. So we compared, our aim was to make recommendations. So in the context of our motivating example, our real clinical trial, we performed a simulation study to compare those different strategies. And we consider different types of scenarios. So in the first scenarios here, we have no effect at all on the whole population and on each group, of course. So none for the phase 2 and none for the phase 3. In scenario 2 here, we have response, the same response in both groups, biomarker negative and biomarker positive. So a global effect in the whole population. And then we have other scenarios, I take this one, where we have no effect for the biomarker negative subgroups, but it's efficient for the biomarker positive. So overall, we have an effect which is diluted here because we are putting together these two groups. But of course, we should select only the biomarker positive. So I will not show you all the numbers, but I will give you the key findings from our simulation. So as maybe we could expect, there is no single strategy which is optimal in all scenarios. It really depends on the scenarios. When you have a broad effect, so when all patients respond, in fact, the best strategy is just to go with your full population and test the full population. But we also noticed that the strategy where you consider testing both in parallel population and subgroups, in fact, perform well, and you have only a small loss of power. And when you have an effect in your subgroups only, subgroup testing or enrichment has a higher power. But you will need, if you want to have good performance, you will need to increase your sample size compared to a trial where there is an effect in the overall population. but still it's a good option. And when it's uncertain, when you really don't know if you may expect an effect in the full population or subgroups, the strategy consisting of testing both is a balanced approach. To note that with adaptive enrichment, there is a potential risk of excluding biomarker negative too early. And if you have an effect in the overall population, you may miss it if you select wrongly only the biomarker positive. And also, it may be more complex to implement. So our recommendation is that you should really guide your decision depending on your clinical evidence and the biomarker data you may have. And then you should choose the strategy based on your confidence in who may benefit. If you really think that the treatment works in the full population, in fact, it's complex and there is no need to consider subgroups and so on. But you may be confident that it works in the full population, then just go with full population. If you're confident that the benefit is only for the subgroups, the biomarker positive, then go only with biomarker positive subgroups. But if you're uncertain, that's where a combined approach may offer a balanced and flexible solution. And of course, you need to do that, ensuring that you maintain your trial integrity. So to conclude, subgroups analysis can enhance clinical trial by identifying patients who benefit most from new treatment drugs. And our study provides evidence for balanced trial design that address both overall and subgroups effects. So we have more precise treatment recommendation for patients. We can reduce risk of exposing non-responder to ineffective treatment. And we can improve outcomes by targeting therapy to responsive subgroups. And we can wonder if future direction maybe should we incorporate biomarkers more systematically in clinical trials? So thank you.